Well, it's my distinct pleasure to introduce Rick Lifton, who's a native of our nation's capital and then went on to Dartmouth University, where he got his bachelor's degree, moved transcontinentally to Stanford University to get his MD and Ph.D. degree in biochemistry, and then moved back transcontinentally to Boston, where at Brigham and Women's Hospital he received his house staff training and then stayed on as a faculty member at Harvard before being lured away to Yale University in 1993. There he's risen through the ranks and is currently the professor and chairman of the Department of Genetics at Yale University. And Rick has done a wonderful job at using very powerful tools of genetics to understand renal disease, and has, I think, taken one of the hypotheses of Arthur Guyton about the kidney being what controls the amount of fluid in that balloon and helps control blood pressure. Rick has taken that and actually shown that hypothesis to be true by demonstrating several human diseases, human examples in which mutations in various transporters or channels and regulators in the kidney are important contributors to changes in blood pressure. So it's a great deal of pleasure that I introduce Rick Lifton.
Thanks, Walter. It's a real pleasure to be here and have the opportunity to speak today. What I thought I would do in the context of this symposium is discuss the natural alliance between genetics and physiology, because I think genetic tools are spectacular at giving us new insights into physiologic pathways and rate-limiting steps that we might have had a very difficult time sorting out from physiologic analysis alone. And so I wanted to develop that theme a little bit in the course of today's discussion. So the notion is that when we don't really know who's the driver and who's the passenger, genetics is a spectacular way of making that decision. Mutations can tell you this by specific mutations can identify key nodes and rate determining steps in complex pathways. And of course, it shouldn't escape our attention that these targets that are identified might be manipulated for health benefit in the general population. So I came into this from a background in Drosophila genetics, and when a generation ago we started trying to understand the basic drivers of development in the fruit fly, we took a simple genetic approach of doing ENU mutagenesis and looking for mutations that had drastic effects on the phenotypes that we were interested in. And this is an example of such an ENU-induced mutation that results in single gene mutation that results in not a subtle effect on body size, but instead a threefold effect on the size of the larva and dramatic effects on the size of the adult. And we're interested in looking for similar effects in the human so that we might identify not places that we might be able to tweak the system a little bit for health benefit, but places that we might be able to have spectacularly large effects that would be important clinically. And I'll give you one example of this. So this is the in vivo equivalent of the Bruce Willis character from the movie Unbreakable. So this is a fellow who suffered a motor vehicle accident in Bridgeport, Connecticut on a Saturday night, was taken to a local emergency room and evaluated. He had routine cervical spine films done to rule out an occult fracture. And the internist who saw him that night was a resident on call, and he said, you know, you don't have any fractures, but I'm very concerned about you because you've got the densest bones I've ever seen. And he was concerned that he might have multiple myeloma. And he referred him up to Yale to see Carl Insonia in the endocrine section. And Carl measured his bone density, and he found that he indeed had the highest bone density on the planet. His lumbosacral spine z-score, the number of standard deviations removed from the mean and standard deviation of the population, was 8 standard deviations out. And if you can find a z-table that goes out that far, it's one with many, many, many zeros on the end of it. But it turned out, when we evaluated this fellow's family, he did have the highest bone density on the planet, but not by that much. Half of his family members had bone densities that rivaled his, whereas the other half were just like anyone else in this room. They were stone cold normal. And this demonstrated that this was a simple autosomal dominant trait transmitted through his family that we were able to map and ultimately identify the underlying gene product. And the mutation in this family turned out to be a single amino acid substitution in LDL receptor-related protein 5, a gene that through Drosophila genetics had been implicated in the Wnt signaling pathway as a co-receptor with frizzled for the Wnt signal. And the mutation in this patient and family was a single amino acid substitution at a position that had been completely conserved in LRP6 homo-orthologs from fruit flies to humans. And when we did biochemistry on this system, we demonstrated the mechanism by which this mutation imparts its effect. It turns out that DKK and sclerostin are normal antagonists of Wnt signaling, and they antagonize Wnt signaling by binding to LRP5 and inducing its clearance from the cell surface through endocytosis through clathrin-coated pits, and that in the presence of these single amino acid substitutions, these patients have sustained Wnt signaling. Now, the beauty of this, I think, is if I had come to you and said, you know a lot about physiology, what is your guess as to what the phenotype of these patients is going to be? They have constitutive gain of function in the Wnt signaling pathway. Those of you who work in development might have said, well, gosh, we know that in Xenopus we can get two-headed tadpoles from gain-of-function mutation in Wnt signaling. Those of you who work in cancer might say, we know a lot of epithelial cancers that have increased activity of the Wnt signaling pathway. So you might be surprised to find that the only clinical complaint these patients have is that they sink when they try to swim. And other than that, they have extraordinarily high bone density, and they're protected from the development of fractures. These bones have extraordinary integrity and suggests that this actually might be a tractable target for therapeutic intervention. Interestingly, in this internet age, once this paper was published, a number of patients self-identified themselves to us. One interesting example was a physician in Alabama called me and he says, I think I might have this trait. And I said, why do you think that? He says, well, I'm 80 years old, I developed arthritis in my hip, and they took me to the operating room for a hip replacement. I woke up after the surgery, I had this big scar on my leg, I was in a little bit of pain, my physician came in and I said, how'd the operation go? He said, we're unable to replace your hip. And I said, why was that? He said, because you broke every drill bit in the operating room. So we've had a number of examples like this, and because of understanding the biochemistry of this rare family, there are now monoclonal antibodies to DKK and sclerostin that are in human trials that are having quite interesting effects, not just to prevent further bone loss, but to promote bone anabolism and formation of bone. So this is one example of the kinds of things you can do with combining genetics and providing insight into basic physiology. As Walter indicated, one of our major interests over the years has been the understanding of the pathogenesis of hypertension, which again is a physiologic conundrum. It affects 1 billion people worldwide and a major public health problem because of its contribution to death from stroke and myocardial infarction. And it's very frustrating that we have a hard time deciding even which organ is the primary cause of this trait. And as Walter indicated, this goes back to the physiologic analysis of the trait. And this is Arthur Guyton's working model for the regulation of arterial blood pressure, devised at a time when we knew perhaps 1/3 of the components that we recognize now. And I think you'll concede this is a very complicated tree. And as a consequence, this looked to us like an ideal place to use genetic approaches to try to understand the trait. And so the notion was, could we find single mutations that would disrupt the overall behavior of this entire trait and drive blood pressure either to the extreme high end of the distribution or instead the extreme low end of the distribution? And of course, we knew we were on to a good idea because so many people in the field told me when we were starting out on this that this was hopeless and would never work, because can't you see that this is such a complex tree that if you place any lesion here, there are so many ways you could compensate that you will never find single lesions that have large effect. Well, fortunately, that turned out to be incorrect. We have identified now mutations in 10 genes that will drive blood pressure to the extreme high end of the distribution, and another 10 that will drive blood pressure to the extreme low end of the distribution in the population. And these genes are not littered randomly across the physiologic landscape, and instead they converge on a single final pathway that regulates how the kidney handles salt. And the simple bottom line is all of the mutations that increase net salt reabsorption by the kidney raise blood pressure dramatically, and mutations that impair salt reabsorption by the kidney dramatically lower blood pressure. There are diverse effects on handling of calcium, magnesium, and potassium. But if you know the vector of what's happening to sodium and chloride, you know what is happening to blood pressure in humans. So these are all single gene Mendelian traits that have been identified by positional cloning, and I'll give you a few examples of these. So this is a diagram of a nephron, and some of the salt transport pathways are indicated. The overall activity of the system is regulated by the renin-angiotensin system, which through angiotensin II causing increased secretion of aldosterone, through its receptor, the mineralocorticoid receptor, a member of the nuclear hormone receptor family, induces increased activity of the salt-retaining pathway. So one example of this identified by David Geller, a fellow in the laboratory, was a single amino acid substitution in the ligand binding domain of the mineralocorticoid receptor that caused a syndrome of severe hypertension that caused profound exacerbation during pregnancy. And the single amino acid substitution was in the ligand binding domain, and by combining structural biology and cell biology and biochemistry, David was able to demonstrate that this single amino acid makes a new van der Waals interaction across the ligand binding domain between helix 5 and helix 3. And this new van der Waals interaction eliminates the requirement for the normal interaction between the 21-hydroxyl group of aldosterone and helix 3. So in the presence of this interaction, you no longer need this interaction, and the consequence of this is that steroids such as progesterone, which normally bind but fail to activate the receptor, are now potent ligands for the receptor. And so women, when they become pregnant who harbor this mutation, progesterone levels go up 100-fold during pregnancy, and they develop rip-roaring pregnancy-induced hypertension. And all women with this mutation have required delivery at about week 30 because of uncontrollable hypertension as a consequence. Just downstream of the mineralocorticoid receptor is one of its major targets, the epithelial sodium channel. And a Mendelian form of hypertension called Little syndrome is caused by mutations that cause constitutive activity of the epithelial sodium channel. So the channel is normally cleared from the cell surface by endocytosis through clathrin-coated pits. There's a typical NPXY domain in the carboxy termini of the epithelial sodium channel that permit its clearance, and mutations in patients that cause either the complete loss of the carboxy terminus or mutate one of 4 amino acids lead to sustained expression of the epithelial sodium channel at the cell surface. They remain active and enter into this same final common pathway of increased salt reabsorption, leading to expanded plasma volume initially, increased cardiac output, and elevated blood pressure. So starting with a blank slate, we now have a host of mutations that do this. On the downside of blood pressure, there are a variety of syndromes that cause life-threatening, typically fatal, hypotension in the neonatal period that all result from impaired salt reabsorption. The most potent place to induce this trait is in the thick ascending limb of Henle, where about 30% of the filtered load of salt is reabsorbed. And we've identified 4 genes that, when mutated, cause this trait called Bartter syndrome. And if you have mutations in the target for furosemides, the sodium-potassium 2-chloride cotransporter, you have a severe salt-wasting phenotype associated with activation of the renin-angiotensin system, induction of hypokalemia and metabolic alkalosis. But you can't supplant this missing channel adequately, and these children frequently die from the severity of their salt wasting. But we can get similar phenotypes from mutations in a chloride channel that the genetics suggested and subsequent physiology has proven mediates the exit of chloride. So chloride that comes in across the apical lumen. It has to get across the basolateral surface to the interstitial space. And loss of function in this specific chloride channel, it results in a phenotype that is clinically almost indistinguishable from that induced by loss of the entry step. But more interestingly, from a therapeutic perspective, we can get a similar phenotype from loss of function mutations in a potassium channel, ROMK, first described by Gerhard Giebisch and cloned initially by Steve Hebert. And loss of function mutations in ROMK give a very similar phenotype with one very intriguing difference. These patients all have marked hypokalemia, typically with potassiums between 1.1 and 2. These patients never develop that level of hypokalemia, and in fact their potassium levels tend to hover between 3 and 3.5 and typically don't require potassium replacement. And the reason for that is this same potassium channel is used in the distal nephron for net potassium secretion. And as a consequence, this has become a very attractive target for development of novel antihypertensive diuretic agents. There are several aspects to this that are interesting. First, these patients with this never develop the same kinds of hypokalemia, which is attractive. Second, this target upregulates in response to treatment. So those of you who are clinically active recognize that when we give furosemide to a patient, the first dose they get a brisk response, the second dose a little bit less response, and then they develop a threshold. You have to keep increasing the dose to get sustained salt wasting. The reason for this is that the target is autoregulated, and if you inhibit it, its gene expression goes up dramatically. That doesn't happen with ROMK, and so for this reason, this actually might turn out to be a good target for antihypertensive agent, whereas this has never turned out to be a particularly attractive target for hypertension and has been more confined to the treatment of congestive heart failure. And these agents are under development in the pharmaceutical industry as well. So I'm frequently asked, what are we going to do when we run out of these rare Mendelian traits? And I'm always reassured that we will find more. And we published a new one just this last month that I'll tell you about briefly because it gets us into new territory. Uda Scholl is a physician who came to the laboratory, and I tell every physician who comes to the lab that there are new traits out there that they should try to recognize. So she went through our database of 1,000 patients with hypokalemia and metabolic alkalosis and identified 5 patients from 4 families who had the phenotype of Gittleman syndrome, which features hypokalemia, metabolic alkalosis, low magnesium levels, and low urinary calcium levels that's attributed to loss of function mutations in the thiazide-sensitive sodium chloride cotransporter. But they had, in addition to that trait, profound neurologic disorders featuring seizures, deafness, ataxia, severe mental retardation, and lower extremity weakness. So this was a new syndrome, which we've called SeSAME to reflect all of the diverse clinical features. And she posited that this might be caused by mutation in a single gene, and in, quite remarkably with the technology we have now, in the span of 2 weeks went from identification of these patients to identification of the underlying gene. And what she found is that all of these families are linked to the same chromosome segment, and they all have loss of function mutations in KCNJ10, potassium channel. And KCNJ10 was previously known to be expressed on the basolateral surface of the distal convoluted tubule of the nephron, and is also expressed in astrocytes in the brain. And we think the mechanism by which these mutations impart the diverse clinical features goes as follows. In the kidney, it turns out that the distal convoluted tubule on a per cell basis is the most active cell in salt reabsorption. And as a consequence, the whole process is driven by the sodium pump, extruding potassium and bringing in, extruding sodium and bringing in potassium. And because this is being done so briskly, in this cell type, salt reabsorption becomes highly dependent on the ability to recycle the potassium that comes into the cell back out across the basolateral surface. And this turns out to be mediated by, at least in part, and dependent upon KCNJ10's gene product, KIR 4.1. It's believed that it's likely also an active heterodimer with KIR 5.1. However, we have not found mutations in KIR 5.1 in any of these families. So we think we can explain the phenotype that looks like Gittleman syndrome because you're stopping salt transport across the epithelium in this cell in a similar way to you're stopping salt reabsorption through the thiazide-sensitive sodium chloride cotransporter. We think the seizures and other neurologic phenotypes come from the absence of spatial buffering by astrocytes. Turns out in astrocytes in the brain, KCNJ10 sets the resting potential and is in close apposition to active synapses, and that KCNJ10 is responsible for clearing potassium from the synapse in response to neuronal activity. And the absence of this spatial buffering, promotes increased activity and results in seizures and the other neurologic phenotypes. So this is the first example of a potassium channel acting on the basolateral surface that is impairing salt reabsorption in the kidney. In the rest of my time, I want to talk about a project that we've been working on for a number of years that continues to interest us and has actually turned us into more physiologists than I'd care to admit. So this starts off with a classic paradox, though, in physiology, and it's a paradox in aldosterone action. The kidney sees aldosterone in two physiologic states. First, if you become dehydrated, you activate the renin-angiotensin system, and this angiotensin II causes the adrenal glomerulosa to increase its secretion of aldosterone, and that promotes salt reabsorption. On the other hand, we know that potassium, hyperkalemia, is a direct secretagogue for aldosterone and causes the kidney to maximize K secretion. And this raises the question, when the kidney sees aldosterone, how does it know whether it ought to be maximizing salt reabsorption or maximizing K secretion? And one of the great things about being at Yale is you have all of these great physiologists that you can go down the hall and ask this question to. And when I asked Gerhard Giebisch and Peter Aronson and Steve Hebert, they all said, well, you know, hemodynamics, everything kind of works out. And that was not a very satisfying answer. And it was particularly perplexing because of this disease, pseudohypoaldosteronism type 2. So these patients have hypertension with hyperkalemia despite normal aldosterone secretion. And it looked to us like these patients behave as though they are stuck in a physiologic state in which they can only do this and can never do this. So this got us very interested in this disease, and Rick Wilson, a really brilliant Ph.D. student, took this problem on, and he positionally cloned the genes that cause this disease. And it turns out that they're caused by a novel family of serine-threonine kinases, the Wnt kinases. And what Rick showed was that mutations in Wnt1 can cause this trait. And the mutations that cause this in Wnt1 are large deletions that take out a big chunk of the first intron of Wnt1 and cause increased expression of its gene product. Conversely, a paralog Wnt4 on chromosome 17 does not have similar mutations, but instead has missense mutations that cause the disease. And these missense mutations are clustered within 4 amino acids distal to the kinase domain of the protein, suggesting that these are going to be some peculiar gain-of-function mutation, because these do not look like simple loss-of-function mutations. So we've had at this point the typical paradox in human genetics, a genotype on one hand, a phenotype on the other, and no idea what connected the genotype to the phenotype. And so we've had to do a lot of work over the years to figure this out. But what has emerged from this is that these Wnt kinases are a previously unrecognized layer of regulation on electrolyte homeostasis in the kidney that are actually orchestrating the behavior of the salt and potassium flux pathways in the kidney. And so Chris Colley, a very talented MD Ph.D. student in the laboratory, has really led this activity. And what he has shown is that Wnt4 normally in its basal state inhibits the activity of the thiazide-sensitive cotransporter, inhibits the activity of the epithelial sodium channel, and inhibits the activity of the potassium channel ROMK. And the single amino acid substitutions in Wnt4 that cause disease in humans have divergent effects on these different pathways. They eliminate inhibition of the thiazide-sensitive cotransporter. They eliminate inhibition of the epithelial sodium channel, but they augment inhibition of the potassium channel ROMK, and they further augment opening of the paracellular pathway for chloride. And the net effect of this is to maximize salt reabsorption and to minimize potassium secretion. So we think we can explain the physiology that we see in patients on the basis of these observations. This got us to thinking that, well, if this is true, perhaps there's a third state of this protein that would actually mediate the converse to maximize K secretion without maximizing potassium secretion. And a very talented undergraduate at Yale, Aaron Ring, who is now an MD PhD student at Stanford, demonstrated that this is in fact the case, that Wnt4 can be phosphorylated in its carboxy terminus by the kinase SGK, and this specific phosphorylation has a different effect from the mutations that cause PHA2, and these alleviate inhibition of the epithelial sodium channel, which provides the driving force which in conjunction with loss of inhibition of ROMK promotes K secretion. And so this suggests that there are three states for this protein, one that a basal state, one that promotes salt reabsorption without K secretion, and then the converse. And most recently, in collaboration with Gerardo Gamba, we've tied this, as we anticipated at the outset, to angiotensin II signaling. That angiotensin II is acting through Wnt4 to achieve this state, suggesting that the mutations that cause disease in humans are a phenocopy of the state induced by angiotensin II. But I have to be mysterious about this because we really don't understand what is going on at the site that is mutated in patients with PHA2 and why this is a phenocopy of what occurs with angiotensin II. So if this all really occurs, it makes a very specific prediction that if we were to make mice that had increased activity of the wild-type Wnt4, it ought to lower blood pressure and lower potassium. And if we do the opposite, if we make mice that had a copy of the PHA2 mutant Wnt4, it ought to raise blood pressure and raise potassium levels. And this turns out to be the case. Maria Laliotti made these back transgenic mice. Gene is expressed under its endogenous physiologic promoter. The mice with an additional copy of wild-type Wnt4 have lower blood pressure than their wild-type litter mates. Mice with the PHA2 mutation have elevated blood pressure, and this is a pair of mice with the different copies of the gene that were pair-raised, showing the differences in blood pressure. The effects on potassium are equally dramatic. Most impressively, if you put the PHA2 mice on a high potassium diet, the mice all die within a day, and if you back off on the K that you put in the diet, you can have mice walking around with potassiums of 8.5. All of this, interestingly, is corrected by giving them thiazide diuretics. And the mechanism for this is interesting and mysterious. We think that the major mechanism of this mutation is to augment activity of the thiazide-sensitive sodium chloride cotransporter. And when you look at these mice, Compared to wild type, this is staining of the distal convoluted tubule and staining for the thiazide-sensitive cotransporter. This is low power and high power. The mice with the additional copy of the wild type gene are hypoplastic for the DCT, whereas there's dramatic hyperplasia of the DCT in mice with just a single amino acid substitution, distinguishing this from this. And the mechanism of this is not entirely clear. So there are 4 of these Wnt kinases in the human genome. 2 of them are mutated in these patients with hypertension, and there are 2 more, and we're interested in what they're doing. And Jesse Reinhardt in the lab has led this effort. And what he found was that Wnt3 has the interesting property that it is an activator in xenopus oocyte expression systems of all of the sodium chloride cotransporters that mediate physiologic chloride entry into cell using the favorable gradient for sodium. And on the other hand, they are inhibitors of all of the KCl cotransporters, which mediate chloride efflux out of cells using the favorable gradient for potassium. So this, of course, raised the question, why would you want a single protein to be regulating these 2 alternatives in reciprocal ways? And we, of course, immediately thought of 2 possibilities. One is cell volume control in response to osmotic stress. So cells are routinely exposed to different osmotic states, and they have to respond acutely in order to avoid either becoming pycnotic or exploding due to water flux across the membrane. And it's known from years of prior work that they do this by acutely modulating their intracellular chloride level. And they do this by modulating the balance between chloride entry and chloride exit. And it's been known from pharmacologic studies that this is mediated through phosphorylation of these proteins. In the phosphorylated state, the entry step for chloride is active, the exit step is inactive, and in contrast, if you dephosphorylate these proteins, the entry step is inactive and the exit step is active, reducing intracellular chloride. There's another state in which this is relevant, and this is in the neuronal response to GABA. As you know, GABA opens a chloride channel, and as a consequence, the resting intracellular chloride level determines the response to GABA. And it's been known for a number of years that in neonates ranging from rodents to humans, GABA is excitatory because the resting intracellular chloride level is high in the neonatal period, and so GABA opens a chloride channel, chloride runs out, and this makes GABA an excitatory neurotransmitter. However, in most nuclei, in most of the brain in the adult, intracellular chloride level is low because the balance is shifted to marked increase in KCl cotransport. So this got us interested in this problem, and Jesse decided to pursue this in more detail. And so he has pursued this by developing and getting up and running quantitative phosphoproteomics in the laboratory. And the way he approached this was to make stable lines that had KCC3, one of the KCl cotransporters, that he introduced at a defined site in HEK cell genome using the Klip recombinase system. And this KCl cotransporter is under TET regulation, so he can turn it on and off. It has a MYC epitope tag on it, so he turns on the protein, expresses it, purifies it by immunoprecipitation, cuts it out of a gel, does tryptic digestion, and then puts it through tandem mass spectrometry in order to identify sites that are phosphorylated. This technology has improved dramatically with the recognition that we can quantitatively enrich phosphopeptides on titanium dioxide columns. And this enabled Jesse to identify 5 phosphorylation sites on the KCl cotransporter shown here. This then led to the question, if you acutely expose these cells to hypotonic conditions, do any of these sites quantitatively change? And in order to do this, he used another new proteomic technology called SILAC, which uses stable heavy isotopes of lysine and arginine to differentially label proteins in different conditions. So you grow your cells in either normal media or these heavy stable isotopes, expose these to hypotonic conditions, these to isotonic conditions, lyse the cells, mix them all together, and repeat the mass spectrometry. And you now have a beautifully internally controlled experiment where you can quantitate the level of these and compare them directly to one another. And compared to the non-phosphorylated proteins, the phosphorylated sites, 3 of them showed no change in response to the hypotonic conditions, and 2 of them showed about a 33% reduction in phosphorylation. And these 2 sites turn out to be interesting because when you mutate these sites individually or together to alanine, you can produce constitutively active KCC3 that is no longer dependent upon exposure to hypotonic conditions for activity. So this shows, this is work in collaboration with Biff Forbush and shows no activity. So these are individual wells and rubidium labeling in response to in response to brief exposure to rubidium. So there's virtually no activity in the wild type. The single amino acid substitution at one of these sites results in about a 5-fold increase in activity. That's equivalent to what we see with the wild type cotransporter in hypotonic conditions. The second site individually has an equivalent response, but you combine these 2 sites together and you get 20-fold induction of activity, constitutive activity that's quite dramatic. There's and just as a control, one of the sites that didn't change, if you mutate that to alanine, has no effect. So these 2 sites that are, have this effect turn out to be completely conserved among all vertebrate KCC3s and also completely conserved among all of the other KCCs 1, 2, and 4. Interestingly, the phosphorylation sites are sites for no known kinase, indicating that there's work to be done to identify the specific kinases that regulate these sites. Jesse has tried to identify the phosphatases, and in vitro phosphatase PPP1 and PPP2A in vitro are capable of dephosphorylating. In the presence of colliculin A, you can antagonize the activation in hypotonic conditions with the wild type cotransporter. The 991A mutation is now insensitive to colliculin A, suggesting that it is the major site that is regulated by this phosphatase. But there are still a number of questions to sort out. We don't know whether dephosphorylation at one site promotes the dephosphorylation directly at the second. We don't know whether the dephosphorylation events are stochastic or not. So does this really happen in vivo? Jesse has gone on to study this in human red blood cells. And in human red blood cells using phosphospecific antibodies, if you expose these cells to hypotonic conditions for 5 minutes, you see dephosphorylation at these 2 regulatory sites, just like you see in the HEK cells, telling you that this is in fact occurring in red cells as well. Another nice technique that has come online with proteomics is the ability not just to say, yes, it's phosphorylated and no, it's not, but to determine the precise stoichiometry using multiple reaction monitoring, using synthetic peptides that are heavy labeled, so you can you can correct for differences in ionization efficiency. So this enabled Jesse first to ask, does phosphorylation regulate whether KCCs get to the membrane or not? And it turns out that it doesn't. If you biotinylate cell surface proteins and purify them, you can see by the antibody staining that they're already highly phosphorylated at these sites. But by MRM, Jesse has been able to determine the absolute stoichiometry of phosphorylation. And in both HEK cells and red cells, the 991 position is at least 90% phosphorylated in both, and the 1048 position is phosphorylated 40 to 65% stoichiometry at both of these sites. So Finally, in the brain, we posited that these sites ought to be phosphorylated in KCC2 in the neonatal brain, and when KCl cotransport is low, and dephosphorylated in the adult brain when KCl cotransport is high, if this model is correct. And it turns out that that's in fact the case, that both of these sites, this just shows the Y and Z plots for one of the sites in mouse KCC2. Both of these sites are phosphorylated in the neonatal mouse brain. We can't do cell surface purification like we can with cells in culture for mouse brain, but overall there's about 20% stoichiometry of phosphorylation at the 906 site in neonatal mouse brain, and this diminishes to undetectable levels by day 21 and persists into the adult. This work is unpublished, but is coming out soon in Cell. So finally, we are interested in trying to tie this to the Wnt kinase pathway and other kinases, because we don't know what kinases are responsible for the phosphorylation at these sites. And so we've begun doing a large screen of RNAi for kinases to see which kinases will knock down phosphorylation at these sites. So far, we've completed analysis for 6 good candidates, SPAC and OSR1, as well as all 4 of the Wnt kinases in HEK cells. So it's quite simple. You expose the cells to RNAi, grow them for a while, then induce expression of the protein briefly, and look for phosphorylation with our phosphospecific antibodies. And to cut to the chase, the only one of these kinases whose knockdown suffices to reduce phosphorylation at these sites is Wnt1. But further work is required to determine how this works. We don't think it's likely that Wnt1 is actually the direct kinase that phosphorylates the sites that we've identified. So a lot of work remains to be done. So what I've tried to show you this morning is how taking relatively unbiased genetic approaches can identify new layers of physiologic regulation that we haven't previously recognized. And what I want to leave you with is the recognition that our genetic tools continue to just get better and better. So I've chaired the oversight of the large-scale sequencing program for NIH, and when we started sequencing the human genome in 1998, cost about $100 to sequence 1,000 bases. And we have beat the heck out of Moore's Law over the last 10 years, because the cost of DNA sequencing has come down 14,000-fold over the last 10 years. And we can now sequence all of the exons in the human genome for about $5,000. And that number is going to come down probably another order of magnitude over the next several years. And when we started talking about a $1,000 genome, it seemed like a fantasy, but I think it's actually going to become a reality much sooner than we might have guessed even a year ago. And as a consequence, the relevance of these kinds of genetic approaches to complex physiology, I think, are going to get better and better. And human as a model system, I think, is only going to get more tractable as we go forward, and I think underscores the utility of thinking about complex problems in human. So I'll stop there, and thanks very much for your attention.
So Rick, thank you very much for an illuminating presentation. We have some questions. Mark?
Going back to your first overview slide, and you suggested that somehow what you were doing was determining rate determining steps. That isn't really very clear to me. If you close the door, obviously you'll stop something happening. It doesn't mean that the door was the rate determining step. And so, I mean, in terms, I think what you've shown is wonderful. I know very little about the details, but the hope of the medical implications. But I would like to take a little bit of an exception in the results that you have for this very complex network that taking one gene does something means necessarily that gene is involved in the rate determining step and further the network in some other complex way.
summarize the question?
Yes. So the point is, if you take one thing out, it doesn't necessarily prove that it's a rate-limiting step, because there may be many different pathways that would do the same thing. And that's a great point. So if I had merely shown you one gene that when mutated caused hypertension, I would have a very hard time convincing anyone in this room that salt reabsorption in the kidney was an important rate-limiting step. However, I didn't show you one. I showed you on the summary slide that taking an unbiased approach and looking for families all over the world, we have identified 10 Mendelian forms of high blood pressure, 10 Mendelian forms of low blood pressure, and we've identified the genes responsible in each of those. And they haven't been scattered randomly across the physiologic landscape. They've instead focused on a single final common pathway that regulates how the kidney handles salt. And so that's how I come to the conclusion that this actually is one of the major pathways that sets long-term blood pressure in human. But I think it's very important, your point is a very important one, and I accept it. It also, however, is worth pointing out that regardless, some of the targets that we identify that have large effects are actually tractable therapeutic targets and are worth pursuing.
What percent of hypertension is accounted for by the genes you identified?
So the question is, what fraction of hypertension is accounted for by the genes we've identified? And this is a wonderful question, because hypertension affects 1 billion people worldwide, and these rare Mendelian forms affect a tiny fraction of the total population. But to address this point as to whether there may be more common variation in these genes that affect blood pressure in the general population, last year we completed a study resequencing some of these genes in 3,000 members of the Framingham Heart Study. It's a study that has longitudinal blood pressure follow-up over a 60-year period. And what we found, so we intentionally picked diseases where recessive loss-of-function mutations cause severe lower blood pressure in these rare patients. And so what we found was that 2% of the Framingham population is heterozygous for these mutations. And these mutations actually have quite large effects on blood pressure, about 10 millimeters of mercury at age 60, which is about what we would get from the use of a single antihypertensive agent. And that, in fact, is sufficient to confer a 60% reduction in the diagnosis of hypertension among the carriers, and further is sufficient to, it appears, out to age 80, there is not a single cardiovascular death and not a single stroke among the mutation carriers in the Framingham cohort. And so there are about 100 million people worldwide who are carrying just those heterozygous mutations in just those 3 genes. And as the cost of DNA sequencing gets lower and lower, I think we'll be able to address these more broadly.
What's your opinion on why there were no mutations identified in genes that we think of that regulate vascular resistance?
Yeah, so I think that's a great question. Why don't we see genes that affect vascular resistance? And I think there are 2 compelling alternatives. The first is really simple. Genetics can't see the effects of mutations if they're lethal. So if these mutations caused embryonic lethality, we would never see them as in human genetics. So I think that's a serious consideration. The other possibility, of course, is that the reason is that if you had primary effects on the vasculature that did not include the kidney would simply adjust the, as we know it does in vivo, would simply adjust its salt handling to return blood pressure to normal. And there have been quite elegant physiologic experiments that have done just this. If you cause increase in systemic vascular resistance by reducing perfusion, for example, to the extremities without affecting perfusion of the kidney, you don't change blood pressure in the long term, despite the fact that you've had a dramatic increase in systemic vascular resistance initially. The kidney adjusts adjusts intravascular volume. And these are experiments that Guyton and colleague and Hall did 30 years ago. And it's quite convincing that the vasculature alone, without the participation of the kidney, is insufficient. On the other hand, if you tie off the renal artery or just impair the renal artery, you can enter into this same final common pathway by activating the renin-angiotensin system, but impairing the kidney's ability to respond to by changing volume. And there are humans who have renal artery stenosis who have hypertension on this basis.
So if we have no further questions, Rick, I'd like to thank you for
Thanks very much.
presentation. Thank you. Now I'd like to introduce George Dugan.
So our next speaker is Dr. Amira Klip from the Hospital for Sick Children and the University of Toronto. Dr. Klip's trajectory is relatively simple. Mexico to Canada to Switzerland and then back to Canada. Throughout all that time, though, she's really focused on a very fundamental problem that has a lot of both physiological and pathological significance. How does insulin regulate the glucose, the transport of glucose into skeletal muscle, which of course everyone knows is the largest depot for glucose. This has a lot of both physiological and pathological significance. But again, as most of you know, the biology behind this is a fundamental area of cell biology: exocytosis, endocytosis, putting a transporter into the membrane and retrieving it. And this has been a problem that's cell biologists, endocrinologists, physiologists have been studying for a long time, but I think Dr. Klip's work has really provided some really fundamental insights into this. Amira.
Thank you, and I'd like to start by congratulating Walter and the department on their new initiation, and I'd like to thank you also for the opportunity to share our work here. I think, just like George said, we're going to move now from hypertension and its ominous prevalence to a disease that also faces us very much as a population and as individuals, and that is diabetes, but I will type 2 diabetes in particular. But I will speak much more about how we understand insulin action, and I'll be happy over question period to tell you where things go wrong. The title I gave to this presentation is Bifurcations in Insulin Action, and if you bear with me through the talk, you'll see why this is not a linear pathway and what we know so far. But what we're going to try to do is get us from the insulin receptor to a molecule called GLUT4, which is the muscle-specific, muscle and fat-specific glucose transporter that is in charge mainly in bringing in glucose during a meal, but also in bringing in glucose when we need to exercise. And regrettably, I won't be able to talk to you about exercise today. So you already have the word there, translocation. And the reason for this is that, again, as George said, this is a molecule that we've known for 25 years does the job, but we've known much less on how it does the job of bringing in glucose. But it involves post-translational cycling. I'll start from this side of the slide. GLUT4 being this red squiggle, it is known that it populates a diversity of still poorly identified compartments. It's not purely the normal constitutive recycling compartment. It segregates away into specialized regions, specialized proteins in those regions. And it cycles, and again, as George said, it rapidly gets endocytosed and very slowly gets externalized. And it's this balance of endo and exocytosis that insulin and exercise-dependent cues and hypoxia have utilized to change acutely the levels of glucose transporters on the membrane so that you can acutely, again, bring in more glucose. And glucose is the main initial source, of course, of energy into our cells. The glucose that gets internalized in response to insulin, its fate, though, is not so much to give us instant energy, but to be stored as glycogen. This is a prediction of how GLUT4 is folded because it is based on other transporters of the same family. But the point to make is that its NNC termini are facing the cytosol. And if this molecule is cycling, you would depend on its regions that face the outside to really tag it when it's outside. And those regions present very few opportunities for epitope tagging. And so us and others have resorted to actually label those exofacial regions. And I'll show you in a moment how that has helped our work. So in addition to the transporter having very few regions to reach from the outside of the cell, skeletal muscle, which as mentioned is the tissue that largely takes up glucose during a meal, is extremely complex. And if you wanted to study either with cell biology tools or with biochemical tools, where in all of these different cell types is GLUT4 in a subcellular level, you would have a really hard time to get to it. So instead, and perhaps a little bit as a cop-out and as a cell biologist, what we use is a muscle cell line that differentiates in culture from myoblasts to myotubes, and that's what muscle cells do normally during muscle regeneration or muscle genesis. And these cells are shown here. Basically, they look simply as your growing monocellular cells. They will form myotubes that in other slides you will see. But we've stably transfected into these cells the GLUT4 molecule I was mentioning to you, in which now there is an inserted MYC epitope that will just facilitate its tagging both biochemically or for immunofluorescence or electron microscopy. So now we have these muscle cells. They, I should say, have all the capacity to respond to insulin in terms of all the signaling elements. We will review them today. And now they're expressing this glucose transporter that will allow us to tag and ask, where is it in time? And we've recently incorporated the same transporter stably into a transgenic mouse in muscle only. So we hope that eventually we will be able to go back to the mature tissue and be able to move away from the cell lines. But for today, let's focus on what these muscle cells look like, the myoblasts that convert to myotubes. The assay I've been mentioning to you that we use to detect the arrival of the transporter at the membrane consists of detecting the MYC epitope. Let me show you two examples. Both of these are of fluorescence microscopy. Here we have a few myoblasts, so magnification of this kind of field, and without permeabilizing them, we're detecting GLUT4 on the surface. And you can barely see anything on the surface when the cells are not stimulated. If you stimulate the cells with insulin, and these images are taken 10 minutes after giving insulin, but within a minute and a half, you can begin to detect GLUT4 on the surface. So it's a very robust response. It's very gratifying to detect it. You can quantify it. And this is another way in which we also look at it, and we like to do it because it allows us to look inside the cell and ask not just when did the transporters arrive, but where were they coming from. So in this case, we've lifted up the myoblasts, and now they look like any regular cell in culture when you're passaging it, and they go spherical for half an hour to an hour. When they are spherical in these cells, if we have previously transfected them transiently in this case with a plasma membrane marker, in this case the GFP-tagged KRAS molecule, then you see the plasma membrane of the cells, and these cells are permeabilized. These were not. These are permeabilized, and in red we're detecting where GLUT4 is. And so two things I want to point out to you. First is that when the cell is not challenged with insulin, GLUT4 is in a nugget that happens to be a perinuclear region. When the cell is stimulated with insulin, GLUT4 first disperses throughout the cytosol into vesicles, and more importantly, it fuses with the membrane, and you can again score this insertion now by the colocalization with the plasma membrane. So these assays are useful to us because they will allow us to test not just physiologically what makes this transition to happen, but also pathologically at what stage is GLUT4 halted when it doesn't get there, when it doesn't get to the membrane. But let's start with the signals that get GLUT4 to the surface. And I think it's been known for about almost 20 years that the insulin receptor, of course, is a tyrosine kinase in itself that has a target. And typically, everybody talks about two targets, the insulin receptor substrates 1 and 2, very homologous molecules. They bind to the receptor that get tyrosine phosphorylated on multiple sites. And I'm sure you all know this. This attracts PI3 kinase, class 1 PI3 kinase, which then exerts a series of signals that we will go over later on. So one of the first things that we wanted to know is, given that both IRS1 and IRS2 are present in muscle cells, they both get phosphorylated, do they do equal jobs? And to approach this, we took advantage of the fact that the muscle cells are very easy to transfect with siRNA oligonucleotides, and in this way, we can knock out specific genes. And you can appreciate here that we can knock out specifically IRS1. This is an siRNA to IRS1. IRS1 blotted goes down, or we can selectively knock out IRS2, and then you can see the IRS2 levels going down. In the graphs below, we've used either IRS1 knocked out cells or IRS2 knockdown cells to inquire what is the response of GLUT4 to insulin? And let's just focus for simplicity at what happens at 5 nanomolar insulin. What we're showing here is the normal response of the cells. This is the value of GLUT4 translocation when the cells have IRS1, and this is what happens when the cells don't have IRS1. You really impair significantly the ability of the muscle cells to bring in glucose transporters to the surface. This was done using the assays I told you before, if the cells have diminished IRS1. But at the same concentrations of insulin, cells that have diminished levels of IRS2 couldn't care less. And through a series of analyses of other outcomes of insulin, we now can confirm that insulin works through IRS1 to lead to GLUT4 translocation and not through IRS2. This is interesting because when we did analyze other outcomes, not just GLUT4 translocation, we found that the mitogenic response to insulin is via IRS2. So right at the first step, there is differentiation of function. There's another important thing about IRS1, of course, and that's that this molecule, but not IRS2, loves to get phosphorylated by a series of serine and threonine kinases, and all of those have one thing in common. They act on many sites, but they are going to lower the ability of the insulin receptor to tyrosine phosphorylate IRS1. So IRS1 not only is the first molecule that receives the insulin signals that will be required to lead you to GLUT4 translocation, but right there, saturated fatty acids, cytokines, and other conditions that may activate any of these kinases shown here has the ability to stop things at the core. Now, today we're going to move downstream of IRS1 and see what are the other signals, therefore, that emanate from IRS1 and PI3 kinase. I mentioned to you that IRS1 binds PI3 kinase. I won't elaborate on that. There are, of course, numerous studies in the literature. But I want to convince you that there are two important branches of insulin signaling downstream of PI3 kinase. And I'm going to paint them all on this graph now, on this slide now, and then we're going to go one step at a time to see how this was demonstrated. So I'm going to try to convince you that downstream of PI3 kinase, RAC1 is GTP loaded, that this, among other things, but presumably because of the change in actin remodeling, is going to change the dynamics of where molecules are in the cell. I'd like to show you a novel player in this signaling, a molecule that binds to remodeled actin, and that's called actinin 4. And on the other side of the story, I'm going to show you that downstream of PI3 kinase, we have the classical activation of AKT through PDK1 and 2. And so we're going to talk about two different modules, the module downstream of RAC and the module involving AKT. There's been a lot of findings, very exciting findings of what comes downstream of AKT. And just for anybody to ask me if atypical PKCs, which are also activated by insulin in some cells, participate in this pathway, I can tell you that although the jury's still out, the majority of the evidence suggests that this might be, if anything, just an outlier and not leading to GLUT4 translocation. But instead, what we know is that this AKT molecule, or PKB, phosphorylates on 4 very important sites a protein called AS160. It stands for AKT substrate of 160,000 molecular weight. And AS160 is a gap RAB, and today I'll tell you how it works on what we think is RAB8 to perhaps interact with a motor protein, and this motor protein interacts also with actinin 4. So we think that not only we are identifying divergences, but also convergences, and how all these steps lead to GLUT4 translocation. So now I need to prove to you that at least some of these steps indeed work as advertised. So first of all, RAC activation. The assay shown here for RAC activation is one that many of you might have used. It consists of pulling down active molecules of RAC, only RAC molecules that contain GTP, by virtue of their binding to a peptide that has affinity for GTP-bound RAC. And so anytime you will see by this RAC pull-down assay a positive signal, it says that RAC is GTP-loaded. You can see here that in response to insulin, at 5 minutes, but again, this can be seen at 1 minute already. In response to insulin, 5 or 10 minutes, you can see RAC activation. The other lanes are controls, and I won't spend time describing them to you. And we have also a positive control that indeed we're looking at a GTP-bound form of RAC. So insulin rapidly activates RAC1. is this important? Well, you can test this in two ways. One of them is you can transfect the cells transiently with either wild-type RAC or dominant negative RAC. And then you can inquire what happens to GLUT4 translocation. Here we're quantifying GLUT4 translocation. Here I'm just showing you an image that is easier to follow. So if we're looking in these 4 images here, what happens to the surface GLUT4? These cells are not permeabilized. This is basal conditions. This is the insulin-stimulated cell, normal response. And I think you can see in this cell that is there in both places, in both squares, there is a cell, but you can't see the surface GLUT4 because it didn't really get increased with insulin, and that's because these cells are expressing dominant negative RAC. And for those of you who care to see the quantification of similar assays, but in monolayers, you do not cause translocation of GLUT4 to the surface when the cells express dominant negative RAC. It's interesting because if you express wild-type RAC, you may, in fact, already potentiate a bit the insulin response. So this suggests that RAC, in some way, is important for GLUT4 translocation. This is another way of doing it. If you knock down RAC at this level of decrease of expression with siRNA, the normal response of GLUT4 going to the surface is also markedly reduced. I don't think I have it shown in this graph, but what RAC, dominant negative RAC, or SIRAC do under the same conditions is they preclude actin remodeling. And I'm sure all of you would have expected that because that's what RAC does. It reduces actin remodeling. In a slide or two, I'll show you what that actin remodeling does. But I first want to bring the question, how do we know that this is either downstream of PI3 kinase or, as we've been painting it, parallel to AKT activation? Well, that it is downstream of PI3 kinase was actually shown here when you look at wortmannin. When you give wortmannin, you markedly reduce, although do not obliterate, the ability of RAC to get GTP loaded. And so we think this gives credence to the fact that, at least to some degree, PI3 kinase is required for RAC activation. There are GEFs for RAC that are PIP3 sensitive. What about independence or dependence on AKT? Well, what you can see here is that, for one thing, we are painting RAC separately from AKT because when we knock down RAC, as is shown in the last 2 lanes of all of these gels, we knock down all the known signaling from RAC, which are shown here for PAC and LIMP kinase, but we do not affect the insulin-dependent phosphorylation of AKT. This is no insulin, this is insulin. And this change is the same as in control cells, yet these cells in the last 2 gels in each box do not have RAC. So we think that this tells us that you do not need RAC for AKT to get phosphorylated. We also have evidence for the converse that I'm just going to tell you about it, and that is that we either use dominant negative mutants of AKT or have used a very effective AKT inhibitor, and under those conditions, RAC couldn't care less. And those are the reasons why we're putting both of them as downstream of PI3 kinase. It's well known, of course, that AKT is downstream of PI3 kinase, as well as in a bifurcation pathway. So let's continue with what's downstream of RAC in terms of leading us to GLUT4 translocation. And I'm not going to focus on these 2 molecules, but rather on a consequence of these and other molecules, that is the remodeling of actin. I've been talking about remodeling of actin. This is what it looks like. In all these micrographs, what you're seeing is only the filamentous actin. These are segments of the myotubes. Myotubes are many hundreds of microns in length. This is a piece of a myotube. These are these multinucleated cells that the muscle cells like to make. Filamentous actin as stress fibers. Insulin-stimulated cells form a mesh in a dorsal region of the cell that is prevented by wortmannin. This would be the same type of response, but in myoblasts, or in these rounded up myoblasts, where you can see the cortical actin forming arborizations in response to insulin that are sensitive to wortmannin. So indeed, insulin causes actin remodeling. All of this remodeling is sensitive to RAC. If I had shown you the dominant negative RAC or SIRAC, you wouldn't have seen any of these manifestations. And as you can imagine, and this is very old work, if you use any of the usual sledgehammer type of approach to prevent actin dynamics, you prevent the gain in surface GLUT4 at the surface. These are cells pretreated with cytokalasin D or latrunculin B, and this is the visuals of it. This is, again, surface GLUT4 in an untreated cell. Barely you can see any insulin response. Pretreatment with latrunculin B, GLUT4 doesn't get to the surface. So I think I've told you, you need RAC to cause actin remodeling. You need RAC to cause GLUT4 translocation. You need actin remodeling to cause GLUT4 translocation. So what's the relationship between GLUT4 and actin? So we collaborated with John Hardwick, and what we're showing here are images of the cortical mesh just beneath the membrane, and that's those big filaments you see throughout. And if you look at the 100 nanometer scale micrograph here, you might be able to see, depending on where you're sitting, black dots, and those black dots are gold-labeled MYC epitopes, gold-labeled antibodies labeling MYC epitopes on GLUT4. And you probably can see that they're all positioned along the filaments. So we think that in some way, the actin mesh just beneath the membrane is at least physically close to where the vesicles are. But of course, we wanted to analyze this relationship between actin and GLUT4 in a more protein meaningful way. And we just heard from Rick about the use of SILAC. So we used the SILAC approach as well, not to look for phosphoproteins or phosphorylated sites, but to look for proteins that may associate with GLUT4 in an insulin-dependent fashion. And just like you heard before, and this was the work of Asaf Rudich and Leonard Foster when they were in my lab, you carry on parallel cultures labeled with leucine or with deuterated leucine, and you can indistinctly stimulate one with insulin or the other. Of course, you need to know which is which. And then you lyse your cultures. You make a single immunoprecipitate of GLUT4. In fact, we immunoprecipitate via that MYC epitope. And you analyze your proteins by mass spectrometry. And the strategy to understand what's going on is that any proteins that are going to have, in this case, more deuterium than hydrogen will be proteins in which insulin has increased their association with GLUT4. Those that don't change don't tell us anything. They might be background, or they might be just proteins that don't change in their association with GLUT4 with insulin. And those in which we have less red signal, in this case, less deuterium than hydrogen, are going to be proteins that insulin decreases their association with GLUT4. So the next table is impossible to see, but I've highlighted the box at the top, which tells us the 3 proteins whose association was increased by insulin. They happen to be cytoskeletal proteins, but not actin, in fact, but proteins such as actinin 4. And we're going to follow up what happens to actinin 4. Interestingly, the proteins that come off with insulin include glycolytic enzymes, and we've also followed that up for other studies that I'll be happy to tell you later. So what is actinin 4, this protein that increases its co-precipitation with GLUT4 in response to insulin? It's a form that, it's a protein that forms homodimers, and in this case, you can see that they have an actin binding domain, and they have spectrin repeats. The actinin 4 dimer is known to mediate membrane to actin binding. So it is a linker between membrane proteins, diverse membrane proteins, including some transporters. And so it was an interesting question for us to ask, is it possible that it binds through its actin binding domain to actin and through what we just demonstrated, the co-precipitation with GLUT4 to GLUT4? And so before going into analyzing this biochemically, we said, do we even care? And so we did what we like to do best, which is let's knock down this protein. But before, I'm just going to show you the co-precipitation of GLUT4 and actinin 4, not just from the mass spec table I showed you that you couldn't see, but rather here through an immunoprecipitation. So just look at the gels. What you can see here is that if we immunoprecipitate via the MYC epitope just look at the myoblasts, the GLUT4, if the cells were not treated with insulin, there's no actinin coming down, but if the cells were pre-treated with insulin, there's actinin 4 coming down with GLUT4. So indeed, biochemically, you can show that GLUT4 and actinin 4 like to come down together. So what happens if you knock down actinin 4? When we knock down actinin 4 to this level, which is about 60 to 70% only of expression knockdown, we obliterate the gain in surface GLUT4 that insulin would cause. So actinin 4 is an important protein for GLUT4 translocation. And I'm going to skip this image here because it says the same thing. So we wanted to know, where is it working? Is it working upstream of actin remodeling? Is it working on the AKT side of things? So the first answer is it's not working on the AKT side of signals, those signals that I had on the far end of the slide. Why? Because when you look at AKT phosphorylation in response to insulin, and I apologize, this is now plus insulin, minus insulin. So this AKT phosphorylation couldn't care less if actinin-4 is expressed or not. So this tells us that whatever actinin-4 is doing to block GLUT4 translocation doesn't involve AKT. And now we ask, is it upstream or downstream of actin remodeling? So what you can see here, it's red may not show very well with the lights, but we'll leave it like that. What you can see here is in these images on this side, these 6 images, in green we have where actinin-4 is. In red we have actin, and in the merged ones, perhaps you can see that when actin is not remodeled, when there is no insulin, the 2 proteins are not together. When actin remodels, actinin follows into the remodeled actin. But more importantly, in cells that do not have actinin-4, and that's why these are black and there's no green signal, actin continues to remodel. I hope you can make out the mesh of actin compared to the basal cells. So this tells us that actinin-4 must be downstream of actin remodeling, because if you eliminate actinin-4, actin still remodels. And you can see that better in these images, where we also ask the question, where is actinin vis-a-vis actin vis-a-vis GLUT4? So we're going to go through these slowly, and I'm just noticing how poor perhaps this is projecting in terms of colors. Oh, that's great. Thank you. This will be perhaps the only time we'll need something like this. That's great. Thank you very much. So let's look at the images just under insulin in each case, in the interest of time. This is, these 3 panels here are in white, but it refers to the localization of actinin-4, actin, and GLUT4 in an insulin-stimulated cell by immunofluorescence. In the regions where actin remodels, you can find actinin-4 and GLUT4. And therefore, when you look at a merged image, everything looks between yellow and white. What happens in cells that do not have actinin-4? Well, now we don't have a signal here. Let's just follow the insulin. Actin still remodels, as I told you before. Look at it compared to a cell that doesn't have insulin. Actin remodels. But there's something very interesting here. Unlike the normal cells where GLUT4 tracked with actin, in the insulin-stimulated cells that don't have actinin-4, GLUT4 doesn't track with actin. And this tells us that potentially actinin-4 allows GLUT4 to be at the same sites as the remodeled actin. That's great. We can go on. So the model we're entertaining is that we have this signaling arm of RAC activation downstream of PI3 kinase leading to actin remodeling, and in some way actinin being downstream of actin remodeling, and I showed you actinin co-precipitates with GLUT4. So we imagine that this arm is important for physical contact of GLUT4-containing vesicles where actin is. So I'm going to take us now to the other side of the question, of the equation, and ask not so much what's upstream of AKT or AKT itself, but I want to tell you about this molecule that you may have heard less of, which is AS160 and whatever is coming downstream of it. So what is AS160? I told you it is this gap for RABs that is a substrate of AKT. That's why it's called, as I said, a AKT substrate of 160,000 molecular weight. It gets phosphorylated by AKT. And being a gap protein, what Linhart and collaborators have hypothesized, and several groups, including ours, have proven to be the case, is that maybe this being a gap, what you need to do is shut it off so that now whatever RAB is its target can prevail in the GTP-bound form. So let me show you what happens if we express an AS160 mutant that cannot get phosphorylated. The hypothesis is that phosphorylating AS160 is going to inhibit its gap activity. I have to tell you, however, nobody has been able to detect the gap activity towards a full-length substrate. So right now, this is all a little bit based on information when you use a small region of AS160 towards its RABs. But let me introduce to you AS160, the molecule. The molecule has all these red marked sites for phosphorylation by AKT. They all get phosphorylated in response to insulin. And what we hypothesized, and Linhart has hypothesized as well, is that these phosphorylations somehow may inhibit the gap domain, so that now you would have a Rab target that would remain in the GTP-bound form if the hypothesis is if AS160 upon phosphorylation can no longer do its gap activity. So how do you prove this? Well, you can do this in two ways. One is you can overexpress an AS160 molecule that cannot be phosphorylated. And in this way, you would eliminate the ability of insulin signaling to shut off the gap. So it would remain always leading Rab towards a GDP-bound form. That bears correct when you do the experiment. I'll show you it in a moment in the next slide. So that would be the hypothesis, as I keep saying, is that insulin, by causing its phosphorylation, would inhibit it, and Rab GTP would prevail. The mutant would not be able to be shut down, and because it wouldn't be phosphorylated, so you would prevail in Rab GTP. The other prediction is that if you do a double mutation, one in which you cannot phosphorylate the protein, but actually the protein was not a gap anyways, that under those conditions, the phosphorylation is not going to do anything. So if you want to think about it, and I tell my group this, think of this protein as a handbrake. If what insulin needs to do is remove the handbrake, but if there was no handbrake to start with, then it doesn't matter whether it can or cannot phosphorylate it. So let's look at the actual result, and this result tells you very basically what happens if you cannot phosphorylate AS160 by overexpressing this mutant that we call AS164P. In reality, it should be called 4A because it has 4 alanines in the sites that get phosphorylated, but that's the name it's gotten. So when you overexpress AS164P, and if you only look at these panels here under surface MYC, this is what happens in the basal state. This is a normal response. This is a normal response to insulin. No surface GLUT4, more surface GLUT4. And if you just look down below, the response is totally obliterated in cells that overexpress the mutant AS160. So you need the phosphorylation of AS160 in order to presumably remove a break and now be able to have GLUT4 translocation. So much for AS160. I'm just going to give you snapshots now of what's known about AS160 and its possible targets. If AS160 is a gap for Rabs, how do you find out which of the many, many Rabs in the cell are required, are its targets? So there were a couple of studies that made some educated guesses. One is, as I mentioned before, that the TBC domain, the gap activity domain of AS160, targets only a handful of Rabs. Those are 2A, 8A, 10, and 14. Now, this is interesting because 2A is in the ER, and we probably can forget about it for the purpose of GLUT4 translocation, at least in terms of establishing a test. So 8A, 10, and 14 were interesting candidates. On the other hand, other investigators have isolated GLUT4-containing membranes from inside the cell and have asked by mass spectrometry what Rabs are present there. And this is the interesting thing. 2A, 4, 8A, 10, 11, and 14 show up. Here underlined are our very same candidates. So then it's a no-brainer. Where are you going to start? And we decided to knock down one at a time, Rab A, 10, or 14. And we also asked, can we, in fact, do a gain-of-function strategy? And I just noted last night that I did not bring the result of this experiment, so I just have to tell you, if you express the 4P mutant, the one I just told you about, if you express that mutant and translocation can no longer proceed because we assume you cannot keep the Rab in the GTP-bound form. However, now you overexpress either wild type or constitutively active Rab 8A or 14, we could rescue with 8A and 14, but not with 10, the translocation. So I'm sorry, I don't have the slide. The slide I do have here for you is what happens if you also try to ask the question independently of AS160. If you knock down Rabs 8A or 10 or 14, what happens to GLUT4 translocation? So here you have evidence that we can knock down selectively 8, 10, or 14. These are controls of signaling. We won't spend time on those. Let's look at what happens here below. All of these are cells that have no insulin. All of these are cells that received insulin. And the bars that did not mount a good response are those from cells that have diminished levels of Rab8A or of RAB14. And regrettably, together, we didn't push things any further. So this tells us that RAB8A and RAB14 independently and sufficiently bring down GLUT4 translocation when they are been eliminated. So this brings us to just the final question in the presentation that I want to give you, and that is, if RABs 8A and 14 are important, what do we know about them? I think I've shown you already that if we inhibit GLUT4 translocation if we knock down RAB8 and 14, just showed you that. I mentioned to you, without the data, that the inhibition by AS160 mutant is rescued by RABs 8A and 14. So what do we know about them? And we're going to focus on RAB8A so far. That's the only one we've analyzed. And I can tell you from the literature, just in the last 2 years, we found out that RAB8A interacts with myosin 5B, and this was done by used to a hybrid system. That made it very interesting because myosin 5B is a motor, and we thought if we now have a motor and we know we have vesicles that bind to actin, perhaps we're beginning to close the loop of the signals and the motors. And that's, of course, what myosin 5B family looks like. These are processive myosin molecules. Myosin 5B, as I mentioned to you, interacts with RAB8, in this case, RAB8A. I'll just show you the last data slide. We tested the possibility that myosin 5B, through its interaction with RAB8A, might be involved in this gain of surface GLUT4 by insulin by expressing a fragment, this fragment or this fragment. These are fragments of myosin 5B. The fragment that is called tail contains a region between amino acids 1221 and 1384, contains a region that binds RAB8A. So this is a fragment that can still bind endogenous RAB8A. This other fragment no longer has the ability to bind RAB8A. And as you can imagine, we did this with the intention to use this fragment to mop up endogenous RAB8A and this fragment as a control. And if you just look at the graph here, I hope you can see that the gain in surface GLUT4 caused by insulin was reduced in cells that express tail, but not reduced in cells that express the other fragment. So we think that this gives us, at least evidence to follow up more on this possibility. And I have to really say this is a model that guides our thinking. I don't think any of these things have been fully confirmed, and probably other ways are required for each step. But I think from what we've done so far, we think that insulin binds to its receptor to activate through IRS1 two downstream cascades, one that includes RAC and actinin, and I'm not sure why here it is. Actinin 4, we think, links to translocation. Very interesting data in the literature, not from us, show that actinin 4 can interact with myosin 5, in fact 5B, which of course is the molecule that we are arriving at through this other cascade, in which I think I showed you that downstream of AKT there's a target protein, AS160, that is a RAB gap, that if you prevent its phosphorylation, you probably prevent a particular RAB getting loaded with GTP. This remains to be measured, but the target RAB that can be shown in vitro to be affected would be RAB8 and a couple of others. And we know from the work I just showed you that RAB8 interacts with myosin 5. It's RAB8A in this case, myosin 5B. And we think this interaction might be important for the translocation because when we express the peptide that interferes with this interaction, and I should say we've shown by fluorescence that we interfere with that interaction, then we abrogate GLUT4 translocation. So this is, in a little bit of a big mouthful, what we think is underlying the movement of vesicles containing glucose transporters to the cell surface. I want to leave you all just with one more word, and that is, what does this have to do with diabetes? Well, type 2 diabetes is preceded by insulin resistance, and you only develop the disease as diabetes with hyperglycemia as its hallmark when the beta cells of the pancreas can no longer resist the insulin resistance. But you can have insulin resistance for 10 years as part of the metabolic syndrome, and that is going to be an absolutely necessary preamble to type 2 diabetes. What is it affecting? Type 2 diabetes, as far as we know today, in all the many cohorts of patients that have been analyzed, and up to 1 in 20 patients has type 2 diabetes in the Western world. No mutations have been identified in any of these molecules, all the way from the insulin receptor to GLUT4 translocation. Yes, we do have, just like in the case of hypertension, selective monogenetic few patients around the world that are affected by a few and not even quite molecules in this pathway. So what is the large number of patients suffering from? These are not, at least not single gene mutations in, that are loss of function of any of these particular molecules, yet the whole signaling pathway is toned down. And I'll be happy over lunch, perhaps that's fitting, to discuss with you how nutrients tone down that signaling. But also, I think it was necessary to have this platform of targets to understand then how our nutrients, cytokines, stressors are going to challenge signaling, because ultimately, insulin resistance of type 2 diabetes is defined as the inability of GLUT4 to get to the membrane in response to the hormone. So I'm going to leave it there. I'm just going to skip this cell biology part, and I'm going to thank the members of my group that have done the work I told you. In particular for today's work, Ilana Talior worked on actinin-4, Tim Chu and Alex Koshkina on AS1 on RAC, Farinder Randhawa on AS160, and the rest of the group are very heavily involved in trying to understand insulin resistance. So thank you very much.
Questions for Rick? Yes.
The microtubules are known to involve in trafficking a variety of molecules. And also in your model shows the myosin 5 involvement in translocating this GLUT4. And this myosin 4 is known to literally walk on the microtubules. So you haven't mentioned the role of these microtubules in translocating this GLUT4 to the cell membrane. And also the second part of my question is, do you think the actomyosin, the actin cytoskeleton roles are limited to stabilizing this GLUT4 at the cell membrane rather than translocating this cytosolic GLUT4 to the cell membrane?
So absolutely, and although this again is a diagram, this conforms exactly the model you're just describing based on data that I don't have time to show you today. I can show them to you later on of where is GLUT4, what is the actin cytoskeleton needed for to bring things or to keep them near the membrane. But if you follow this cartoon, this is based on work from different labs in our own. We believe that the vesicles are going up and down on microtubules, perhaps in an idle way. And we think that myosin 5B, as shown here, is myosin 1C, 5A, and 5B. We think that myosin 5B allows this switch of tracks, precisely as you were mentioning, from microtubules to actin. This is based on other things that are known in the literature about myosin 5B. And we think this is under the control of signaling through RAB8A. So the signaling to RAB8 may allow myosin 5 to really bind on the vesicles and make them change tracks. You need to prove this in a mechanistic way. We think that at the same time, in response to insulin, RAC is causing the actin remodeling near the surface. Actinin-4 is in that actin mesh, and we think this is going to help perhaps stabilize or position or guide GLUT4 near the membrane, because the next step has to be to find productive snare complex formation between VAMP2, which we know is on the vesicle, and syntaxin-4 and SNAP23 on the membrane for final fusion. We think that perhaps signaling downstream of AS160, not through RAB8, but perhaps RAB14, we don't know, might be involved in the fusion. This is based on work from other groups that find that AS160 is important as much for the fusion. Our work that I can show you later shows that it is important that myosin is probably involved in the stabilization near actin, and that RAC is required for that retention. So just like you described, that's our way of thinking.
Questions? Is there any evidence that any of the insulin resistance promoting cytokines can target any of these downstream proteins? I mean, most of it's focused on IRS proteins.
Absolutely. I'm glad you asked, because the literature will largely focus on IRS1, and you will almost find any study looking at insulin resistance showing you that AKT is reduced by about 50% in almost any model, whether it's cytokine-induced insulin resistance, fatty acid-induced insulin resistance. And yet, if you deliberately inhibit AKT only by half, which is what is found in all those studies, nothing happens to GLUT4 translocation. This arm is extremely resilient. So we've done a series of studies using cytokines, using saturated fatty acids, ceramides. And what we found is that actually RAC is exquisitely susceptible. Under conditions where IRS1 binding of PI3 kinase is normal, phosphorylation of IRS1 is normal, we find RAC GTP loading being quite susceptible. So we believe that, if nothing else, it's an arm that is important to pay attention to. And also, it's an exciting arm because if it has a protein that may interact directly with GLUT4, it might give us some ability to improve such interactions. But you're totally right. Every paper in the literature that looks at insulin resistance from the point of view of signaling says IRS1 phosphorylation is abnormal, AKT phosphorylation is abnormal. This is never abnormal by more than 50%, and that's inconsequential.
So you're obviously doing a beautiful job of dissecting the GLUT4 translocation pathway, but you alluded at the end to the observation that we haven't found many genes in the genome-wide association studies that contribute to insulin resistance as a causation of diabetes. And I'm wondering how you're thinking about insulin resistance, whether you think it's contributing to diabetes or whether you think it's actually adaptive in response to the overfed state.
Yes, so it's an excellent question. So if you bear with me for 2 minutes and you still are happy to go for lunch, this is the current thinking, and it's not really my particular view, but I do think there's a lot of truth in it. Obesity-linked type 2 diabetes has a large component of cytokine assault to the muscle, and this whole pathway is very much cytokine sensitive. Where are those cytokines coming from? They're coming from macrophages largely that are going to remodel and then the adipose tissue, and in the course of it, they get activated into the M1 mode, and they start to spill out inflammatory cytokines. So inflammatory cytokines and fatty acids together affect signaling. So this has nothing to do with specific genes. It has to do with specific nutrients. It's overnutrition, as you were mentioning. That's in the case of obesity-induced insulin resistance. Now, obesity does have, and very beautiful work from Steve O'Reilly and others has shown, as I'm sure you know, obesity does track with certain genes. Some very few ones are monogenes, and the other ones are, of course, more complex traits. So our ability to consume energy, our ability to expend energy through exercise, through fat oxidation, all of those are probably targets. So it's really fine-tuning of metabolism that is probably altered in all these individuals that are going to have high levels of fatty acids and high levels of cytokines. And that's going to, as you said, secondarily act on insulin resistance. So do we know of primary genes causing insulin resistance? No, not yet in any case. Do we know, is this secondary as an adaptation? The insulin resistance is happening as a result of the overfed state. It is a preamble to type 2 diabetes. But when the gene studies have been done to find genes that lead to susceptibility to type 2 diabetes, all of them point to beta cell targets. Logically, you're defining the disease as the moment when the beta cell fails. It doesn't say, you need a preamble of insulin resistance.
So this raises the question of if you could dramatically increase GLUT4 translocation in patients, the typical obese diabetic patient, do you think that would be beneficial, or do you think that would be maladaptive?
Well, in animal models, it has been shown that you can overcome insulin resistance caused by beta cell attack. So it's not a model of type 2, but it's for the same reason as in type 2. So if you use streptozotocin or ligate the pancreas, et cetera, in mice that overexpress GLUT4, you prevent the rise in glycemia. And the reason for this is the beta cell gets involved because the insulin resistance exerts high demand. If you don't have that high demand, you're not going to have the beta cell involvement. So I would argue that this would be beneficial. Where there are a lot of targets in the chemical trials right now, in clinical trials, are of chemicals that increase fatty acid oxidation, as I'm sure you know. So AMPK agonists, because by increasing fatty acid oxidation within limit, you're going to reduce the circulating levels of fatty acids. So probably at any level there's an opportunity to intervene, but it's more metabolic.
Any other questions? with that, we'll thank Dr. Klip, and think about that for lunch.